Okay, so welcome back. We are here tonight to do the lecture for the immune system. And you're going to see that the lymphatic system is included in this lecture because the lymphatic system has a large immune function. So oftentimes it depends on what anatomy textbook you're working from, but some anatomy textbooks will lump the lymphatic system in with the immune system. So you're going to see that we're going to talk about those together tonight. So real quick, I'm just going to go ahead and turn on my pen like usual, and that way then we will be all ready to go. I think I'm going to try, how about we'll try green tonight, see how green works on here. Oh, maybe not green. Sorry, excuse me for a second while I play with the pen colors here. How about, nope. Trying to get something that might show up for you guys. I wish they had some brighter colors here for you, but how about we'll just try blue. Hopefully blue. There we go. That one seems to show up pretty well. Okay, so now on to the lecture. So we're going to, again, start off with the lymphatic system. And then we'll get into kind of the more traditional immune system and their immune system functions. But remember that the lymphatic system is going to have a large immune function, and we'll talk about why here in just a minute. So, like the cardiovascular system, the lymphatic system is going to be a network of vessels. But these vessels are going to be transporting things. They're basically going to be transporting things one way. So your lymphatic system is going to be a one-way trip back to the subclavian veins, as you'll see in just a minute. And the main function of the lymphatic system is to transport body fluids. And as it transports those body fluids, it's going to clean those body fluids out of any bacteria or any potential foreign invaders. And you'll see that it'll do that in these places like the lymph nodes and in some of the lymphatic organs like the spleen. Lymphatic vessels, the main function of them again are going to be to collect and carry away excess fluid that builds up in the tissues, in these what we call the interstitial spaces. The interstitial spaces are just the spaces in between cells. So if, let's say here's some muscle tissue, okay? And again, you're going to have, your muscle cells packed in there. But there's going to be spaces in between the muscle cells. And those spaces in between the muscle cells are filled in with fluid, okay? And so that's what we're calling your interstitial spaces. Your interstitial spaces are the spaces in between the cells. And there's going to be fluid in there. And so our lymphatic vessels will pick up this excess fluid. And if you remember, you have blood vessels that are running through here, right? And remember that when we did the blood lecture, we talked about the fact that hydrostatic pressure is going to push fluid out into the tissues. And then osmotic pressure, especially on the venule, if this is a capillary bed on the venule side, osmotic pressure will pull some of that fluid back in. But remember that there's going to be a net movement of fluid out. So more fluid, okay, is leaving due to hydrostatic pressure than is coming back in, okay? So again, if we could draw, and this is just a good chance for me to review, so I hope you don't mind, I'm just going to digress for a minute and review. But remember that, here's our capillary bed here, okay? And here, again, it branches into these little tiny capillaries. That's what I'm trying to draw. Here's your arterial end. And then, again, that arterial branches into these little tiny capillaries. And then these capillaries come back together. So here would be all the fluid traveling through there. And the capillaries then come back together here at the venule end. This is a horrible diagram, but so what happens in this capillary bed? So what happens in this capillary bed, and I'm just going to draw it a straight tube, but remember, a capillary bed is really branched. And so what's going to happen here in the capillaries in this capillary bed is on the arterial end, hydrostatic pressure is going to be really high. Okay, so this is hydrostatic pressure. And that hydrostatic pressure is going to push lots of fluid out. Okay. Now, as you move through that capillary bed and you get closer to the venule side, so this would be the arterial side over here, and this is the venule side over here. As we move closer towards the venule side, then hydrostatic pressure is going to get less and less because we're losing fluid, right? So it's like losing fluid from the hose. And so as fluid builds up out here, we have less fluid inside here pushing out. So on the venule side, we hit a point where osmotic pressure becomes greater than the hydrostatic pressure. And the osmotic pressure is going to suck some of that fluid back in, but not as much, okay, as what originally went out. So if you look, that's why I drew this arrow smaller, okay? So we're going to lose more fluid due to hydrostatic pressure than we are going to gain fluid back in due to osmotic pressure, okay? So what that means is we have a net movement of fluid out here. Okay, and that means that we're going to get some fluid that builds up in this tissue. And so, so that fluid that builds up in that tissue, again, you know, you're going to, over time, build up lots of fluid in that tissue. And some of that fluid, again, is going to get absorbed by the cells. But you're still going to have excess of that fluid in the tissues. And so what picks up that excess fluid so you don't continue to swell is the lymphatic system, lymphatic vessels. And actually what's interesting is, to give you an idea of how important the lymphatic system is, if let's say you have surgery or you had damage to your tissues and lymphatic vessels were cut or damaged, then you can get what's called edema. And edema is the buildup of fluid in your tissues because the lymph vessels that were damaged can't properly drain that excess fluid off. So that's how important the lymphatic system is. Okay, so that was kind of a long introduction, but hopefully it gave you a little review for the blood as well. And so those are your main lymphatic vessels. You also have these special lymphatic vessels called lacteals. And these lacteals are going to be associated with the digestive system and especially the intestines. And they're going to transport fats into the circulatory system. So hence, they're actually called lacteals because the fluid in them looks kind of milky. So that's where they get the lacteal name from. And that milky fluid it looks milky because of all the fat in it. So it's basically lymph fluid with lots of fat. And it transports some of those fats to the circulatory system, and then the circulatory system will transport those fats out to, again, tissues that need them. So the organs of the lymphatic system, we're going to also talk about, and this is just kind of the introduction, so we're going to get into the nitty-gritties on each of these. But the organs of the lymphatic system are going to also, as I kind of suggested, be partially there to help defend the body against disease. And so they're going to have this immune role, and we'll talk about especially the spleen having an immune role and the lymph nodes having an immune role as well. So again, this is kind of my gingerbread diagram of the circulatory system, and then they even included in here the lymphatic system. So of course I had to include my gingerbread anatomy diagram here because I was so impressed that they included the lymphatic system. So the lymphatic system is usually drawn in as like a green color. It's kind of a yellow, more yellow color here, but that's your lymphatic system. Okay, so Here is kind of a nice diagram showing you some of the lymphatic system, and I like this one because it shows you the place for the lymphatic system. So again, remember that the lymphatic system is going to start in the tissues, and remember it's a one-way system. So it's going to pick up fluid from the tissues and dump it back into the circulatory system. So it's a nice way of recycling that excess fluid. The excess fluid that makes its way into the tissues will then be picked up by the lymphatic system and taken back to the circulatory system. So again, the body is just so amazing in that it's so efficient. And again, here's an example of how it recycles that excess fluid. So what happens is in the tissues, there are these lymphatic capillaries, and I'll show you a picture of what they look like. But again, there are these, they don't look like the capillaries of the circulatory system, because remember, the capillaries of the circulatory system are going to connect venules to arterioles. Here now, in the lymphatic system, they're just going to be these one-way capillaries that are going to be kind of capped off. So they're going to look like this in the tissues because, again, they're just absorbing. They're not passing fluid on. They're just absorbing fluid. So they're going to absorb the excess fluid in the tissues, okay? And those lymphatic capillaries are then going to pass on the fluid. So they'll branch into these larger lymphatic vessels. And then the lymphatic vessels, as you can see here, are going to pass through lymph nodes. So that's what they drew here. You got your lymphatic capillaries branching into your lymphatic vessels, and then they'll pass through a lymph node, okay? And then they'll pass into a lymph vessel. And so here's again back into a lymphatic vessel. And then those lymphatic vessels, okay, will eventually branch into your lymphatic trunks. And then your lymphatic trunks will eventually dump into these collecting ducts. And these collecting ducts, you have two main collecting ducts. You have the right collecting duct, and we'll talk about the right lymphatic duct, and we'll talk about this in just a minute here when I show you where it's draining fluid from. So the right lymphatic duct is going to drain fluid basically from the right side of the upper body, so the right side of the arm, the right side of the chest, and the right side of the head. And then the thoracic duct is actually going to drain fluid from all of the rest of the body. So if you look here, this is the thoracic duct right here dumping into the left subclavian. And the thoracic duct is actually going to carry fluid from the lower half of the body, so both the right and left limbs, and then the left side, the abdominal region, and then the left chest region, the left arm, and the left side of the head. So the thoracic duct is a major duct. And so then the right lymphatic duct and the thoracic duct are going to dump into the right subclavian vein and the left subclavian vein. Again, obviously the right lymphatic duct is going to dump into the right subclavian vein, and the left, or the thoracic duct, is going to dump into the left subclavian vein. And that's how the fluid makes its way back to the blood. So that's what they're showing you here. Those collecting ducts are going to dump into the subclavian veins and bring that fluid back to the blood. Okay, so then it can get recycled, reused. So that's the major lymphatic pathway. And again, here I just want you to know kind of the order of how the lymph fluid would move through. So if you had a question like this, hint on an exam, I would basically have you choose from what pathway was the correct pathway for lymph fluid to move through the lymphatic system. So, and this would be one of your answers, and of course this would be the correct one. So now let's go back and look a little bit closer at lymphatic capillaries. So lymphatic capillaries are going to be these tiny closed-end tubes that are going to extend, remember, into these interstitial spaces, the spaces between the cells of your tissues. And they're going to obviously take in that excess tissue fluid through their thin walls, and they have these little one-way valves on them that they'll take that fluid in. And once inside, that tissue fluid is now called lymph, okay? So once the tissue fluid that from the interstitial spaces is absorbed into those lymphatic capillaries, now we call that fluid lymph officially. So this is a really cool diagram. I love this diagram because it's such a good visual of how the lymphatic system is going to absorb fluid. So you can see the lymphatic capillaries again are going to be all throughout the tissues that have your regular blood capillaries, and again they're going to absorb that extra fluid. So as fluid again moves from the blood into the tissues and builds up in the tissues, some of that excess fluid will be absorbed by these lymphatic capillaries and brought back into these lymphatic vessels, okay, and then eventually back into the blood via the subclavian vein. So, lymphatic vessels, when we look at these lymphatic, and this is a really good picture of, this would be a lymphatic capillary right here. And the walls of your lymph vessels are actually thinner than those of your veins. Remember, your vein wall, the walls of your veins are thinner than arterioles, so, or arteries. So, then lymphatic vessels, the walls are even thinner than veins. And they're typically constructed with the same kind of three-layer system. Remember, we had the tunica externa, the tunica media, and the tunica interna. So again, we're going to have those same basic three layers. But now they're going to have these, what we call semilunar valves on the inside. And these semilunar valves are going to be these little one-way valves that let fluid in, okay? Again, they're one way. And so what happens is when fluid builds up in the tissues in the interstitial spaces out here, then that fluid is going to push on that valve, and it'll push that valve open and allow that fluid in, okay? But once that fluid's inside, that's a one-way valve, so it can't get back out. Larger lymphatic vessels will pass through, again, your lymph nodes and eventually merge to form these lymphatic trunks. And those lymphatic trunks will eventually merge into lymphatic ducts. So, again, this is just that same diagram. It's kind of a cool diagram to show you how lymph flows. So, again, you've got your lymph capillaries that are going to flow into your lymphatic vessels that oftentimes go through a lymph node, again, that branch into larger lymphatic vessels that are going to then branch. So again, these lymphatic vessels here are this, really the same type of vessel. It's just that they're branching into larger and larger and larger lymphatic vessels. So you have a lymph vessel here. It moves through a node and then into more lymph vessels, branching into more and more lymph vessels. And these lymph vessels are eventually, again, going to branch into your lymphatic trunks, which are eventually going to dump into your right and your thoracic collecting ducts, and then those are going to dump into your right and left subclavian vein. So, and again, kind of cool because you can see then how the lymph system is dumping that fluid back in. So really, if you want to kind of think of the lymph system in a very basic term, the lymph system picks up the excess fluid, okay, cleans it in the lymph nodes, and then dumps it back into the blood. So now this is kind of cool because this shows you the two major collecting ducts. So we have the thoracic collecting duct on this side and the right lymphatic duct on this side. And so what you're going to see here is in red, here this is all of this tissue here in the area in red. So again, all along the right arm here, and then the right side of the head, and then the right side of the chest region. All of that fluid is going to drain via the lymph vessels, okay, into the right lymphatic duct. So all of the lymph from this region here is going to drain into the right lymphatic duct and then eventually back into the blood via the right subclavian vein. Okay. Now the lymphatic fluid from both lower limbs, the right and left lower limbs, the abdominal region, and then the left side of the chest, left side of the head, and the left side of the arm here are all going to drain into the thoracic duct. And the thoracic duct, again, will drain into the left subclavian vein. Okay. So again, just keep in mind that the thoracic duct is actually draining both limbs from both sides, okay, the right and left. Hint, hint, if you had a question on the exam that asked about, tracing lymphatic fluid from, the left leg all the way back to the blood, then you would have to make sure that you included that it went through the thoracic duct and then through the left subclavian vein, okay, to get back to the blood. So that covers some of the, and this is kind of cool because this actually shows you some of your lymphatic system here, and it shows you that right lymphatic duct dumping into the right subclavian vein. So that's kind of cool. Okay. So, and again, this is just another, I put lots of diagrams here so that you could see in lots of different ways how, again, that lymph makes it back to the blood. So again, here is just another diagram, and if it works for you, then, go with this. I'm a visual person, so I include lots of visual aids. So here's your lymphatic capillaries, again, draining into these lymphatic vessels that are going to move through these lymph nodes. Okay, and then these lymph vessels are eventually going to come together to join a lymph trunk, and then the lymphatic trunks will come together to join a lymphatic collecting duct. Either, again, the right lymphatic duct or the thoracic duct. And then you would, again, enter the back to the venous system. That fluid would enter back into the venous system through the either right subclavian vein or the left subclavian vein. Again, if it was coming from the right lymphatic duct, then that's going to drain into the right subclavian vein. If it's coming from the thoracic duct, then that's going to drain into the left subclavian vein. Okay, so hopefully that makes tracing the lymphatic fluid fairly easy here. So now we're going to talk a little bit about the tissue fluid formation and lymph, and we'll get into then lymph nodes and the spleen and their role and function in the lymphatic system. So lymph formation, again, depends on tissue fluid formation. And tissue fluid is going to be made up of water, of course, and then dissolved substances that are dissolved in that tissue fluid that have left blood capillaries by filtration and diffusion. So remember, that's how we get this fluid built up in the interstitial spaces surrounding these cells, is that, again, we've ultimately, from the capillaries, had a net movement of fluid, as we just talked about a little while ago, net movement of fluid out of the capillaries into the tissues. And so again, here's my tissues, and you'll have that fluid building up in the interstitial spaces here. And then the lymphatic system is to pick up that excess fluid. Wow, my diagrams are getting worse and worse. I apologize. That one's pretty bad. This was supposed to be a lymph capillary here. But anyway, hopefully you guys are able to decipher my horrible drawings. I have no artistic capabilities whatsoever, as you can see. Filtration from plasma, again, normally exceeds reabsorption, just like we talked about. So this is what leads to that excess formation of tissue. So all that's saying is that, again, remember that hydrostatic pressure is going to be greater than, so fluid loss due to hydrostatic pressure is going to be greater than the fluid gain due to osmotic pressure in the capillaries. So we're going to get a net movement of fluid out into the tissues. And again, that excess fluid will eventually need to be reabsorbed by my horrible depiction of a lymphatic capillary here. Okay, and so that's exactly what this is showing again. And this is just showing that as fluid moves out of the capillaries into the interstitial spaces in the tissue surrounding the capillaries, then that excess fluid, as that excess fluid builds up out here, it'll push again on these little one-way valves here, and as it pushes on those nice one-way valves, then again that fluid will enter the vessel and become lymph. So again, it's the hydrostatic pressure, okay? Literally, again, that pressure of the fluid that's building up and building up and building up in the tissue, and when it builds up enough pressure, it'll push open those lymphatic valves and drive the entry of that fluid into the lymphatic vessels or into the lymphatic capillaries, and then it becomes lymph, okay? Once again, remember that fluid moves from the tissues into the lymphatic vessel. It's now called lymph. So now, the forces that move that lymphatic fluid through the lymph vessels are going to be the same ones that move blood in your veins. So you may say, well, how does now the lymphatic fluid get pushed? Because there's no pump, right? Like there's no heart pumping it, the lymphatic fluid, through the lymph vessels. So we have to rely on other forces to move that lymphatic fluid back up towards the subclavian veins. So we rely on a skeletal muscle contraction, just like we did for the veins. So as we contract again the muscles in the legs, for example, and this is why they tell you on an airplane again to remember to move your legs. You want to contract those muscles in your legs. And that way then, as you contract those muscles, they squeeze on those lymphatic vessels, pushing the fluid farther up in those lymphatic vessels towards the subclavian veins. Also, just simply breathing. Breathing in the thoracic and abdominal cavities will compress lymphatic vessels in those regions and help to push that fluid, that lymphatic fluid, up towards the subclavian veins. And then you also have contraction of some of the smooth muscle in the actual walls. Remember, the tunica media of the walls of these vessels is going to contain smooth muscle, and that smooth muscle can be contracted to help propel some of that lymph fluid through those lymph vessels. So those are the main, again, forces that move lymph fluid through the lymph vessels, or skeletal muscle contraction, breathing movements, and again, the smooth muscle, contraction of the smooth muscle in the walls of the lymphatic vessels. Now, a condition that interferes with the flow of lymph results in something that we call edema. And so what that means is if, for example, there were damage to some of the lymphatic vessels, like during surgery, so maybe some of the lymphatic vessels or tissues were removed or disturbed, then we no longer have those lymph vessels and lymph capillaries to pick up that excess fluid. And so that excess fluid can build up in the tissues and result in what we call edema. Okay, so edema is just the swelling or excess fluid that would build up in the tissues. Okay, so lymph nodes. Now let's get to some other structures in the lymph system that we haven't talked about. We haven't talked about lymph nodes. Lymph nodes, as you probably well know, are involved with your kind of defense against invaders. So they're, again, they have this immune function. And so and you guys probably know that because you probably have been sick and had swollen lymph nodes. And so one of the reasons that you get swollen lymph nodes when you're sick is that your lymph nodes contain white blood cells. So they actually contain lymphocytes. And as you're going to find out, they're going to store some of your T lymphocytes and your B lymphocytes in the node. They also are going to have these engulfing cells called macrophages. And so they do include these immune cells, lymphocytes and macrophages. And we're going to talk, don't worry, we're going to talk more about what lymphocytes and macrophages do here as we get into the immune system. But they do have these two immune cells. And so these immune cells are basically going to be out there performing immune surveillance. And So basically, as fluid's being moved back as lymph fluid is moving through there and moving back from the tissues to ultimately the subclavian veins, they're going to clean that fluid, but they're also going to be doing a little surveillance, saying, whoa, wait a second, we got some bacteria coming from, the tissue in the lower legs. And so as they perform that surveillance, they can kind of alert other parts of the immune system to a potential infection. So, and this is just showing you how oftentimes what it looks like in terms of vessels bringing lymph fluid into a lymph node. So oftentimes you have more vessels bringing in fluid into the lymph node than are leaving here. And so that basically creates kind of a backup as we'll build up fluid in the lymph node, and then it kind of forces it through this lymph node like a filter. And so we have our afferent lymphatic vessels, which are bringing, again, fluid into the lymph node. And then we have our efferent lymph vessels that are bringing fluid out of the lymph node or exiting. I always think of efferent as exit, okay? So they're the vessels that are exiting the lymph node. And again, throughout the lymph node here, we have immune cells like B and T cells, as well as macrophages that are going to help kind of filter out any foreign invaders. So we also have B cells in here as well. So again, functions, we kind of talked about this, but functions of the lymph nodes. Macrophages and lymphocytes within those lymph nodes are going to filter the lymph and remove any bacteria, any extracellular debris. So again, cells that are broken down and there's just parts of them floating around. They'll remove that cellular debris. They'll remove bacteria. So that way then that lymph is returned to the blood as this clean lymph. Lymph nodes are also, again, going to be the centers of lymphocyte production, and you're going to see that the cells in this area, especially when we talk about your B cells and your T cells, are going to function in your immune surveillance. So again, they're just kind of keeping an eye on what's coming through that lymph. So, for example, if bacteria are moving in that lymph fluid, if your lymph node here, and all of a sudden you are picking up there are bacteria in this lymph node, that means that there are probably bacteria in the tissue those lymphatic vessels were actually draining. And so what happens is the immune cells here are going to kind of alert the immune system and put the immune system on high alert to look for these other invaders in the rest of the body. So, and here, this is kind of cool. This is a picture to show you most of the major regions where you're going to find your lymph nodes. So you can see, again, in your inguinal region, you have lots of inguinal lymph nodes. Again, in your axillary region, you have lots of lymph nodes. You've got lymph nodes, again, all along the abdominal lymphatic ducts and trunks, and or lymphatic vessels and lymphatic trunks here. And then, again, you, of course, have some lymph nodes, especially in the cervical neck region, in the thoracic region as well. So it just shows you where some of your kind of major clumping of lymph nodes are. And again, you probably have noticed, especially it's easy to notice the cervical nodes here. It's easy to notice that they're enlarged when you're sick. Oftentimes, again, that's because of the excess the immune cells. What will happen, and we'll talk about this when we get into the immune system, is when you have an infection, the immune cells will divide and produce lots of cells, basically, again, kind of building up its army, if you want to say, put it in those kind of analogy terms. It basically is building up its army to fight off whatever your foreign invader is. So that's why your lymph nodes swell. It's those T cells and those B cells and those macrophages that are building up inside that lymph node. So it's actually a good thing when your lymph nodes swell when you're sick. Okay, so the thymus and the spleen are going to be two other major lymphatic organs. And the functions of the thymus and the spleen are going to be similar to those of the lymph nodes. The thymus is going to have a very special function in that it's going to be responsible for containing lymphocytes that will mature into T cells, into T lymphocytes. So it's really responsible for spitting out your T cells. The thymus, just is this soft bilobed organ. It's located right behind the sternum. And let me see if I have a picture here. There we go. Here's the thymus gland right here, just to show you. Here's your thymus. And so again, it's right behind the sternum there. And it's bilobed. It has these two big lobes to it. And it shrinks in size throughout our lifetime. So it's much larger in children, and then it becomes much smaller as we get older. And it contains, again, lymphocytes in it, and this is one of the reasons why, again, as we get older, our immune functions start to become depleted. So our thymus is going to be much smaller, and that means it's going to contain fewer lymphocytes, and then ultimately fewer T cells. So, but anyway, back to our thymus. Our thymus, again, is going to receive these T lymphocytes, and you'll see these T lymphocytes are initially going to be made from stem cells in the marrow. And so some of those lymphocytes that were made in the marrow will move into the thymus, or move through the thymus and sit in the thymus, and in the thymus they'll actually mature into T lymphocytes or T cells. Okay, and the thymus also is going to secrete a hormone called thymosin, and this hormone is one of the major hormones that's responsible for taking just our lymphocytes, okay, that have been undifferentiated, and encouraging them to mature into T cells or T lymphocytes. Okay, so thymosin is our major hormone responsible for causing lymphocytes to mature into T cells. So the easy way to remember this is that T cells, okay, are named after where they mature. T cells mature in the thymus. Okay? When we get to B cells, we're going to talk about B cells actually mature in the bone marrow. Okay, so they're going to stay in the red marrow and mature in the bone marrow. So if you ever wondered where we get the name T cells and B cells, this is where. T cells, again, have matured in the thymus, so TT, and then B cells have matured in the bone marrow. So I know, you guys, see, now you guys can thank me when you win, that double Jeopardy question for, where do we get the name T cells and B cells, right? You guys are thinking, no, that's not enough. But if you do end up winning double Jeopardy, you're going to have to come back and thank me. So, okay, let's get on to the spleen now. The spleen lies in the upper left abdominal cavity. Okay, so again, kind of important to know where some of these major organs exist, right? And if you want an idea of, you've got your spleen in your upper left abdominal cavity, and in the right upper abdominal cavity is going to be your liver. Your liver is going to push up into the right abdominal cavity. But we're just going to focus on the spleen here. So the spleen is the body's largest lymphatic organ. Okay, so the spleen is a lymphatic organ, and the spleen is going to resemble a really large lymph node, except that it's going to contain blood instead of lymph. So again, it's basically going to act as a filter, but again, it's going to filter the blood instead of just filtering the lymph. So inside the spleen, you're going to contain what's called the white pulp. And so here, I'm just going to outline. Here's the white pulp. Okay, if we were to take a cross section of the spleen, there's the white pulp, and the white pulp contains lots of lymphocytes. Okay. So again, we're going to have T cells and we're going to have B cells. And they're just sitting there. Again, remember that in the thymus is actually responsible for putting out T cells. It's allowing them to mature and become T cells in the thymus. Here in the spleen isn't doing any of that. The spleen is just storing them. So T cells can move from the thymus into the white pulp of the spleen or into a lymph node. And so just like a lymph node, the spleen is just going to store these T cells and these B cells so that they're ready to attack anything that's, coming through as we're filtering the blood. So, we've got, again, the white pulp that contains lymphocytes, and then we have the red pulp and the red pulp is going to be all of this stuff. So all of this area right here is going to be red pulp, all here, okay, around here, if you can see that. That's all red pulp. And it contains, again, red blood cells, macrophages, and lymphocytes. And so, again, it's also, again, helping to filter out any of the bacteria or any damaged cells, any damaged blood cells. So the spleen literally just acts as a filter, filters the blood. It's the main function for the spleen, is it acts as a filter. And it's going to remove any damaged blood cells, and it's going to remove any bacteria. So, you can live without your spleen. I actually have a friend who, unfortunately, when she was a child, she fell off a horse, and the horse reared up and came down on her. And when it did, the horse came right down on her left upper abdominal region, and so it damaged her spleen. And they actually had to do a splenectomy and remove her spleen. And so she's lived without a spleen. However, she does know that her immune system is a little bit compromised because of that, and so she did tend to get sick a little bit more often. But other than that, you can live without a spleen. But of course, it used to be that if you had a bruised spleen that they would just remove it, but now they're finding that the spleen actually, you will get some tissue repair in the spleen. So now they're actually opting to leave the spleen in to see if it repairs itself if there wasn't too much extensive damage, because they now recognize the spleen has this immune role. So again, it's helping to filter. It also, again, is filtering the blood because it's taking out some of the old red blood cells that are damaged. So it does have a function. So again, you can see again what happens is you have the splenic artery that brings in, again, blood through the spleen, and then that blood will be filtered, okay, and then it'll move back out the splenic vein. So again, obviously, we're bringing oxygen to the tissues of the spleen as well, but while it's doing that, it's cleaning out any debris and bacteria. So fluid will literally move out of these little splenic capillaries, again, into the red pulp, and then eventually will be absorbed back up into these splenic capillaries on the venule side. And then it's a little more than you need to know, but I was just kind of trying to explain how this happens. So for it to actually filter out what's in the fluid actually, believe it or not, moves out of, again, the arterial end of the capillary into the red pulp, and then is filtered by these immune cells and some of these cells that are going to pick up the old dead red blood cells. And then now that fluid will make its way back into the capillary end on the venule side, and then back into the veins and out. So, but again, it's more than you have to know. I just want you to know the basics on the spleen. You don't need to know how it moves through. The fluid moves from the splenic arteries out into the spleen. I just kind of wanted to explain that because I know some of you like to know how things work. So now we're going to talk about more of just kind of what we would typically kind of consider the traditional immune system, even though, again, oftentimes the lymphatic system is included in the immune system. And so now we're going to talk about our body's main defenses against infection and how the immune system is involved in this, especially, particularly our immune cells. And so again, the lymphatic system is going to oftentimes contain organs that contain these immune cells. So now we're going to get into these immune cells. So disease-causing agents typically in our body are called pathogens. So when we call something pathogenic, that means that it has the potential to cause disease or, again, harm inside the body. And so remember that not all bacteria are pathogenic. Obviously, you have plenty of bacteria that are what we consider are good bacteria, or natural flora bacteria, that are very beneficial for the body. We have bacteria that help us break down things in the digestive tract. We have bacteria that help on our skin, help to outcompete bad bacteria so that it makes it harder for bad bacteria to enter the body. So we have lots of good bacteria in the body. But the bacterial invaders of the body that have the potential to cause disease are the ones that we call pathogens. And they can produce, again, infections within the body. So let's talk about how the body does this. The body has, to prevent infection, the body has two lines of defense against pathogens. It has what we call the non-specific defenses, okay, that will guard the body against pathogens, and it has these specific defenses, or what we call our immunity, that will mount a response against very specific types of pathogens. So our non-specific defenses will basically guard the body against any foreign invading pathogen. And our specific defenses are going to mount a response against a very specific pathogen, so a certain bacteria or a certain virus. So now we're going to talk a little bit more about the innate, non-specific first line of defense, and then we'll eventually get into our second line of defense that protects us, again, against some of these specific pathogens. So let's do our non-specific defense first. And so, first off, one of our defenses that we have is we have what's called species resistance. Each species is resistant to or affected by certain pathogens that are specific to it. So what that means is, obviously there are certain pathogens that will infect us that won't infect dogs. Okay, so a dog is resistant to certain pathogens because simply those pathogens can't infect its cells because its cells are different than our cells. So same thing, we're not affected by all of the same viruses that might affect a dog or a cat because, again, as a species, our cells are going to be different enough that they may not be able to be infected with that certain type of pathogen. So that's what species resistance is. All it says is that as a species, so as a human, there are certain pathogens that infect us, and there are certain pathogens that can't because we're human. So that's species resistance, okay? Then our main first line of defense is going to be mechanical barriers, okay? Mechanical barriers are typically going to be just, our skin and our mucous membranes that help to prevent bacterial invaders from making it farther into the body or, viruses or whatnot. So again, unbroken skin, our skin provides a good mechanical physical barrier, right, to bacteria. Also our mucous membranes, so we have mucus membranes that line openings to the body, like, again, the nose and the mouth. And that mucus it's sticky and it can trap, again, bacteria and viruses as well. And so that's an example of our first line of defense. It's non-specific, right? Okay, it's a non-specific defense. Now, this is going to be considered, and I'm breaking this down a little bit more than this initial chart. This calls your adaptive, the second line of defense. We're going to call our we're going to have our second line of defense that's internal but non-specific, okay? So this is going to be your internal non-specific portion of your immune system. And that is that we have chemical barriers, okay? Again, now this is obviously produced internally, hence why it's internal, but it's non-specific. So that means it'll just kill all bacteria, all viruses. It's not specific for a certain one. And gastric juice is one of them. So it's so funny to me that we always worry about what we take into our mouth. You know, if you're like, oh, I don't want to drink after that person because they're sick, which is good, you shouldn't. But oftentimes, again, those areas, especially taking things into the digestive tract, are usually safer areas because in the stomach we release these digestive juices that are very, very acidic and oftentimes, again, kill a good portion of your bacteria. Now, in your tears, in your eyes and your nose, those are areas where you're relying mostly on mucosal membranes, and so they're definitely much more vulnerable. So your eyes and your nose are areas that you want to be careful with. And I always tell students, especially this time of year, when things are going around, try not to rub your nose and try not to rub your eyes, because your nose is relying mostly on mucosal membranes to trap bacteria, and your eyes are relying on, again, mucosal membranes, and the eyes also produce some things called lysozymes. Lysozymes will lyse bacteria, and they're produced in your tears. It's an enzyme produced in your tears. But again, still more vulnerable than, again, having to, survive the digestive juices. Digestive juices are so acidic. And so, again, this, even with the lysozymes, your eyes are more vulnerable, again, to taking in pathogens. But we do have lysozymes in your tears, okay? Also, part of your internal non-specific defense is interferons. And interferons are basically an antiviral substance. They're produced by cells when a cell is infected with a virus. Okay, and so let's say here we have a cell and it's infected with virus inside. And what this cell will then do is it will produce these interferons, okay, which we abbreviate IFN. So it produces these interferons that then will induce nearby cells. So nearby cells can pick up these interferons, okay? And now it tells that nearby cell to produce these antiviral enzymes. And so that way then, if this cell here is infected with this same virus, okay, it has these enzymes already. They're ready to lyse or kill that virus. So that's the cool part about interferons. Interferons are basically a substance released by an infected cell to tell other cells to basically arm themselves against the virus. So they tell these other cells to produce these antiviral enzymes so they can arm themselves against the virus. Fever, okay? We oftentimes think of fever as being a negative thing, but honestly, a fever is part of your natural immune defense. What a fever is, guys, is basically, if you think about it, your body's smart. It says, hey, look, this bacteria has invaded my body because I obviously provide the right conditions for it to grow. So let's create some conditions that are unfavorable for it. If it likes this body temperature, that's why it invaded me, then let's turn up the heat. And that's exactly what a fever is doing, is it's producing a less favorable environment for that bacteria or for that virus. So fever offers some powerful protection against infection. And again, it does that by basically providing an environment that's less hospitable for that bacteria or virus. So the other interesting thing about a fever in terms of preventing bacterial growth is that during a fever, the amount of iron in the blood is actually reduced, and many of your bacteria require iron to divide and grow. So you're basically now providing fewer nutrients for the growth of that bacteria. So again, that's another benefit of a fever. A third benefit of a fever is that phagocytic cells, so these cells that we're going to talk about, especially your macrophages that are engulfing and attacking these bacteria and viruses, are going to attack with greater vigor when we have a fever, when temperatures are higher. So basically it enhances our attack cells. Okay? So honestly, you know, when I get sick, I very rarely take anything to reduce my fever. I usually honestly just let my fever, unless of course, you know, my fever is starting to creep up, you know, over 102 into the 103 range, then, you know, you're just uncomfortable. And so oftentimes I'll take an NSAID or something like that to bring my fever down. But and for the most part, it's very rare that I will take anything when I have a fever, because remember that fever is part of your natural immune response. And I do the same thing with my son. You know, I try not to give him baby Tylenol if he has a low-grade fever, because your fever is doing all these things. Again, it's providing a less hospitable environment for bacterial growth. It's removing some of the that iron from the blood, and it's providing fewer nutrients for the bacteria to grow. And then it's enhancing our attack cells, our phagocytic cells. So there's lots of benefits to fever, believe it or not. Okay, inflammation is another one of our internal non-specific defenses. So inflammation is in response to a pathogen in our tissues. And so again, for example, if you got a cut and you introduce some bacteria there, oftentimes we would see inflammation there. And it's typically characterized by some redness, some swelling, heat, and sometimes a little bit of pain. And so the major actions that occur during inflammation are, first off, okay, again, if you have a foreign invader, bacteria in these tissues, maybe let's say you had a cut in the skin here. Okay, here's a cut in the skin. And so you introduce some bacteria here, that cut in that skin. So the first thing that's going to happen is the blood vessels in this area are going to dilate. Okay, and what that's going to do is that's going to increase the amount of blood volume in that area. So as the blood vessel dilates, right, and gets bigger in size, more blood can move through that vessel. And again, that is going to bring more blood cells, especially more white blood cells to the area. So then the next step will be that white blood cells will actually invade the infected area. They'll actually move out or emigrate out of, just like you're seeing here, of the blood vessel into the surrounding tissue through this process that we call diapedesis. In just a minute, we'll talk about this, diapedesis. Okay, so those white blood cells will move out into the infected area, and then we'll start to see the appearance of fibroblasts, which are going to be these connective tissue cells, and they'll start to kind of form a sac around the area to help contain the bacteria. So this is a nice diagram right here of what you would typically see in inflammation. So again, first off, you're going to have the blood vessels dilate. And as those blood vessels dilate, guys, it's bringing more blood volume. And so it's bringing more fluid into that area. So you're going to get a little buildup of fluid in your tissues as well. And that's one of the reasons why you see swelling. But the good part of that is that, again, with that extra fluid, it's bringing in more white blood cells. And so more white blood cells can move to that area and take care of the bacteria or whatever invader you have in that area. And so again, here you can see white blood cells are going to move out of the blood vessels into the surrounding tissue through this process called diapedesis. They kind of squeeze out. And then once out into the tissues, they will actually move towards the area where the bacteria are through a process that we call chemotaxis. They'll actually be drawn to certain chemicals produced by the bacteria and other immune cells in that area, and they'll move towards that area through this process that we call chemotaxis. So phagocytosis is another, again, our phagocytes are another internal non-specific. These are all non-specific defenses, okay? So phagocytosis just means that we have these active phagocytes, specifically neutrophils. Do you remember neutrophils and monocytes? We talked about these when we talked about the blood chapter, and these were some of your white blood cells. So now we're connecting again the dots back to one of our previous lectures. So remember that neutrophils are what we called our bacterial slayers, right? And we also have monocytes that can eat bacteria, but monocytes will actually attack larger things as well. And so both neutrophils and monocytes are going to leave the bloodstream at areas of injury through this process called diapedesis that I just mentioned. Diapedesis just is the process by which white blood cells are going to squeeze out of the blood vessel, okay? We call that diapedesis. All it means is these white blood cells squeezing out of the blood vessel. And so both neutrophils and monocytes will do that at the site of injury. Remember that neutrophils are primarily going to be our bacterial slayers. So they're engulfing small particles, especially bacteria. Okay. Monocytes are typically going to attack larger ones. So monocytes will actually oftentimes attack entire cells that are infected. So for example, if one of our cells is infected by a bacteria, or if it's infected by a virus, then a monocyte will engulf the entire cell. So monocyte typically attacks larger ones. It can also engulf parasites. So sometimes they'll engulf some of our larger parasites like parasitic roundworms or something like that. Okay, so both monocytes and neutrophils, though, will engulf things in a similar manner, and they're going to do it through this process that we call phagocytosis. And you guys probably remember phagocytosis from the beginning of the semester. So what happens is, initially, the microbe, whether it be, a bacteria or some type of parasite, will adhere to either the monocyte or the neutrophil, and then what will happen is the phagocytic cell will produce these little foot-like structures that are going to stick out. We call them pseudopods, and those pseudopods will stick out and eventually kind of surround continue to push out until they surround the microbe invader. And then eventually they'll actually push out, and then eventually they'll just pinch off here. They'll just come together and then they'll just pinch off right here. And when they pinch off, now you have your microbe trapped inside a vesicle. Okay? So that's what it would look like if that pinched off there. And now we have that microbe entrapped in a vesicle. And so now the benefit of this is, now that it's trapped inside this vesicle, we can fuse this vesicle here with a lysosome. And the vesicle containing the microbe, we oftentimes call a phagosome. So now the phagosome can fuse with a lysosome. Now we call it a phagolysosome, okay? And remember that lysosomes contain enzymes that digest stuff. So now these enzymes will digest the bacteria, okay, and kill it off. And then what we'll have left is this, basically the residual body, the leftover undigested parts. And then those undigested parts can then be, removed from the cell through exocytosis. So that's how, again, some of these phagocytes will basically break down bacteria, and they'll break down parasites. So far, guys, we've talked only about our innate defenses, okay? These are our innate defenses. So you're going to see some books are going to call this your first line of defense, second line of defense, and then this is your adaptive ones are your third line of defense. It just depends on the textbook, but I'm going to use the term innate defenses and adaptive defenses because that's one that you seem to see is pretty consistent. So your innate defenses are going to include your surface barriers, your skin and your mucous membranes, as well as some of your internal defenses, your phagocytes that we talked about, like the monocytes and the neutrophils, fever. We didn't talk about natural killer cells just because your book doesn't focus on them as much, but we also have these natural killer cells that are part of the immune system that will help to kill off bacteria and things like that. We have antimicrobial proteins that we'll talk about in a little bit, and inflammation. And so those are all our innate, non-specific. We call that our innate, non-specific defense. Okay. Then we have our adaptive defenses, and that's what we're going to talk about now. And our adaptive defenses are going to include humoral immunity and cellular immunity. Okay. So our B cells and our T cells are going to be our adaptive defense, and these are going to be specific. This defense is specific for a certain type of invader. So let's talk about our specific defense or immunity. And again, oftentimes you're going to hear this called the body's third line of defense. Okay, and so this third line of defense is going to refer to our responses that are mounted by the body, against a very specific invader, so a certain bacteria or a specific virus. Okay. So now we have to talk a little bit about what antigens are, and we've kind of talked about antigens before when we talked about ABO blood typing, but we were very general there. So when we talk about antigens, antigens are any type of protein, identifying protein on a cell that identifies that cell as being whatever that cell is. So for example, if you are a certain Streptococcus aureus, that's a specific species of bacteria. If you're a Streptococcus aureus bacteria, okay, then you are going to have very specific proteins on the surface of your cell that are going to identify you as a Streptococcus aureus bacteria, okay? Or again, if you are another type of bacteria, Staphylococcus, bacteria, then you're going to have certain identifying proteins that will identify you as that type of bacteria, okay? We obviously in our cells have antigens that identify our cells as self cells to us, okay? So antigens are basically these little ID tags, okay, that are on the cell that identifies a certain cell as being a certain type of cell. So when we talk about antigens in our body, we can talk about self or foreign. We consider antigens in our body to be foreign. Like if we're using the term antigen, then it has the capability of eliciting an immune response. And so typically we refer to them as foreign. But we do have, obviously, self-antigens, meaning that we have proteins on our cells that aren't eliciting an immune response in our body, but are eliciting an immune response if we put them in someone else's body. Okay? So, again, that's all that antigens are, these little proteins on the surface of cells that act as little ID tags. And they are, again, molecules that can potentially elicit an immune response. Okay? So, now let's talk about, and if you look at this picture here, you're going to see that they've got antibodies. And antibodies will be very specific. Each antibody will be very specific to a certain type of antigen. So now we're going to talk about how we actually produce antibodies. And so that's where we have to talk about lymphocytes. Okay? So lymphocytes during fetal development are going to be released from the red bone marrow. And from the red bone marrow, they're released into circulation, and about 70 to 80% of those lymphocytes will mature into T cells in the thymus. Okay? The remainder of the lymphocytes will actually go back to the bone marrow and become B cells, okay, in the bone marrow. So that's where we get T cells and B cells, right? T cells are going to be lymphocytes that have traveled to the thymus and matured in the thymus to become T cells. And B cells are lymphocytes that matured in the bone marrow, and hence the name B lymphocytes or B cells. Okay, so our T cells and our B cells are our two major cells that are important for our adaptive immunity or our third line of defense. And our T cells are going to be, again, this reminds you, they mature in the thymus. Our T cells are going to be involved with what's called our cell-mediated response. We'll talk about this in just a minute. And our B cells in the bone marrow are going to be associated with our antibody-mediated response. So I want you to think, when you think of antibodies, think of B cells, okay? Antibodies are going to come from B cells. Both B and T cells are activated in your secondary lymph tissues, so in your lymph nodes and your spleen and your lymph nodules. So what this means is, again, lymphocytes are going to be made in the marrow, right? Okay. Some of those lymphocytes, or most, I should say, of those lymphocytes are going to travel to the thymus and mature into T cells. And then some of them will stay in the marrow and become mature B cells. And then B cells and T cells will then travel through the circulatory system into your lymph nodes, into your spleen, and into lymphatic nodules, and they'll hang out there and act again as these surveillance cells ready to attack. So again, undifferentiated lymphocytes, we already kind of talked about this, sorry that this is a review here, but I got ahead of myself. So undifferentiated lymphocytes that reach the thymus, again, become T cells. B cells that stay in the bone marrow will mature again, or lymphocytes that stay in the bone marrow will mature into B cells. Both B and T cells are going to reside again in lymphatic organs. So once mature, they'll move to the lymph nodes or the spleen or lymphatic nodules. So this is kind of a cool diagram because it shows that. So it says, again, stem cells in the red bone marrow give rise to undifferentiated lymphocytes. Then the lymphocytes will move through the blood, and some of those undifferentiated lymphocytes will move through the thymus and mature in the thymus and become T cells. Some of them will stay within the bone marrow and become B cells. And then, lastly, both the T cells and the B cells are transported through the blood to lymph organs, such as the lymph nodes, lymphatic ducts, spleen, lymph nodules, where they're going to hang out in those lymphatic organs ready to attack. Okay, so now let's get into some of the lymphocyte functions here. T cells will attack foreign antigen-bearing cells, especially bacteria, by what we call direct cell-to-cell contact. This is what we call cell-mediated immunity. Okay, again, these T cells, and I'll show you a picture of this in just a minute, are going to be directly attacking these foreign antigen invaders, so especially things like bacteria. T cells can also secrete these certain cytokines. These are cellular chemicals. They're kind of like they almost act like hormones, except for, again, they can be released into the tissues. They don't have to be released into the blood. So they're basically these cellular signals that we call lymphokines. And these lymphokines will actually enhance the B cell response to antigens. So T cells help activate the B cells, basically, okay, through these lymphokines. Okay, so let's talk a little bit about, again, these T cells. So this is an example of some of our T cells, and we have really two types of T cells here. These are going to be what we call our cytotoxic T cells, okay, the ones that are actually going to directly attack bacteria, and these ones that are releasing the cytokines are going to be our helper T cells, okay, because they're helping to activate B cells. So in this picture here, okay, this is showing our cytotoxic T cells. And so what these cytotoxic T cells will do is they can either directly attack bacteria, or they may actually bind to an infected body cell that's displaying one of those antigens on the surface of it. And then what it will do is it will release these destructive enzymes that will actually kill off the cell and all the bacteria inside of it. So again, these cytotoxic T cells can actually not only directly destroy bacteria, but they can also directly destroy cells that are infected with bacteria. B cells will attack pathogens by differentiating into plasma cells and secreting antibodies. So B cells are going to attack, guys, in a much more indirect method, okay? And so remember that cytotoxic T cells are going to be very direct. That's what we call cell-mediated immunity. Here, this is going to be called B cells are going to be involved with what's called our humoral immunity, because what a B cell will do is a B cell will not directly attack an infected cell. What a B cell will do is a B cell, okay, will produce antibodies, then release antibodies from the cell, okay, that will travel through the blood, hence the name humoral, right? Because blood's one of the humors. So the antibodies will travel through the blood and then again to the infected cell, where then the antibodies will basically bind to and now tag that infected cell for destruction. Okay? So let's say this cell here was infected. Okay, and when a cell is infected, what will happen is the cell will then display the antigen of that, let's say it's a bacteria. Okay, there's a bacteria in here that has this antigen. Okay, so now the cell will actually display that antigen. And so what will happen is if we've produced antibodies to it and the antibodies are released into the blood, then the antibodies can bind the cell, and now they've tagged it for destruction. Okay, so now this cell is marked for destruction. A lot of people think that antibodies are directly attacking the bacteria or the infected cell, and they're not. They're actually just tagging it for destruction. It's kind of like giving the kiss of death, you know? I'm giving my mob analogies now. I'm not sure why, but you know, if you were talking about, you know, all the mobster movies out there, right? And you know, you've got the head boss, and he oftentimes doesn't do the killing. He just gives the kind of kiss of death and tags that person, right? So that's what the antibodies are doing. Is they basically are the kiss of death, and they are flagging that cell now for destruction, so that other cells like, again, your macrophages, which are one of your monocytes, will come in and engulf that cell now. Okay, so that's basically what they're saying. Body fluids, again, attack and destroy specific antigens or antigen-bearing particles through antibody-mediated immunity, also called humoral immunity. And that's because, again, these antibodies have to travel through the blood. Okay. So we'll talk a little bit more about how this happens. I'll kind of go over that process again here in just a minute. So T cells and the cellular immune response. T cell activation, again, is going to require the presence of a cell that will present antigen to it. So what this means is, and this is what you see here, okay? Here we have an infected cell that's displaying antigen on its surface. This cell is what we call an antigen presenting cell, an APC, because it's presenting antigen, okay? So we need some type of cell to present antigen to the T cell to activate it. So sometimes it can be a B cell, sometimes it can be a macrophage. Again, sometimes it can be an infected cell that has already encountered the antigen. And so when that happens, then we will either activate our helper T cells, okay, or our cytotoxic T cells, or some of those cells, okay, will become memory T cells. So when we activate a T cell, especially something like a helper T cell, oftentimes that T cell can undergo division and produce some memory T cells and then some helper T cells, okay? So here we've got a T cell that was just activated, and it will undergo cell division and produce some memory cells, okay, that'll hang out in our system to remember that specific antigen for later if we're reinfected. And then we also are going to have our T helper cells. Our T helper cells can go ahead and directly now activate B cells. Or again, that T cell may divide and produce cytotoxic T cells. And those cytotoxic T cells, again, can go and directly kill an infected cell. B cells and the humoral immune response. So a B cell can become activated, and when a B cell becomes activated, okay, it is going to produce a clone of cells, okay? So what's going to happen is, here's this B cell, okay, and let's say it was presented with antigen. When it's presented with antigen, it will undergo cell division and produce a clone of cells, okay? Most of these B cells, okay, are going to, when it divides, are going to become plasma cells. And these plasma cells, okay, their main function is just to pump out antibodies, okay? So all of these plasma cells are going to make tons of antibodies and then release antibodies, okay? So that's the main function of plasma cells, is they are producers of antibodies. But some of those B cells, okay, here, will become memory B cells. And that way then, they'll hang out in our system in case we are reinfected with that same bacteria. And then instead of, just having this one B cell have to undergo cell divisions, we'll have all of these memory B cells out there ready to undergo cell divisions if we're reinfected, so that our immune response will be much greater the second time that we're infected with that antigen. So, again, now keep in mind that most of your B cells are going to require a T helper cell to activate them. So most of your B cells need a helper T cell to become activated. So what that means is, T cells are important for helping to activate our B cells and our antibody response. So they're kind of interrelated. So this is kind of cool because this shows you that we have these different B cells that are out there floating around in our system. And our B cell will have these little receptors on the surface that are going to be very specific to certain types of antigen. So, for example, here's our antigen floating around, and here's all our different B cells. And so in this case, this B cell here actually is activated because it's specific for this type of antigen. So that B cell binds that antigen, and then now this specific B cell, okay, will undergo cell division and produce lots of clones of itself, okay? And some of these clones will mature into plasma cells. Actually, most of them will mature into plasma cells. And the plasma cells, if you look, what makes the plasma cell a little bit different than this B cell floating around is that it is going to have tons of endoplasmic reticulum, guys. And the reason that it's going to have all that endoplasmic reticulum is it has to produce tons of protein, right? And remember that the endoplasmic reticulum produces proteins that will leave the cell via exocytosis. So that's exactly what they're doing. It's producing tons of the antibodies, which are proteins. So it's producing tons of those antibodies and then releasing those antibodies from the cell. Okay, so that's what your plasma cells are doing. And then, of course, some of the cells, as they undergo cell division here, are going to become memory cells. And those memory cells will stay around and hang out in the immune system, especially in your lymphatic organs, like your spleen and your lymph nodes, so that it's ready for the next attack. Okay, so this is kind of cool. I found this because I found that students seem to have a lot of trouble with picturing what's going on with B cells. So what I did was I found this great little cartoon about B cells and even helper T cells. So hopefully this helps you. Here is a B cell, okay, specific for a certain type of antigen. And so here it says the B cell finds an antigen which matches its receptors. So here's your B cell. Oh, this one fits. Okay, so then that B cell oftentimes has to wait until it's activated by a helper T cell. So helper T cell will come along and activate that B cell. So here in this case, here's your helper T cell activating your B cell. Now, once the helper T cell activates that B cell will start to divide. And that B cell will produce, as it divides, some of those cells will become plasma cells, and some of them will become memory cells. Now, the plasma cells here are going to produce antibodies. So you can see it spitting out antibodies, right? And then the antibodies will go and attach to the invader and tag that invader for destruction. And then your eater cells, which are going to be your phagocytic cells, okay, your phagocytic cells like your macrophages, are going to come along and they will engulf that cell that was tagged for destruction. So in this case, a bacteria. And then you have your memory cells, and your memory cells will stay inside, again, certain lymph nodes and spleen and things like that. And if the same intruder, that same bacteria invades again, then the memory cells help the immune system to activate much faster because you have more of them out floating around. So when you have that invader that invades the system for a second time, now you have more of these B cells ready to be activated. And so if you're activating more of these B cells, then as though all those B cells undergo cell division, they're putting out more plasma cells and more antibodies than your first response. Okay, now let's talk about antibodies. So again, we'll talk about, remember those plasma cells that spit out the antibodies. We'll talk about the different types of antibodies they can spit out. So there are 5 major types of antibodies, or immunoglobulins, that constitute what we call the immunoglobulin fraction of plasma. And so remember that these antibodies, when the cells spit them out, will be picked up into the bloodstream and then will make their way through the bloodstream to the area of infection. So, but we have 5 major different types of antibodies that are involved with different types of things. And I apologize because this is on 2 slides, and when you make your note cards, just combine some of this information. I meant to combine these slides, and I didn't get a chance to. Let me go backwards. Sorry. Whoa, I jumped way ahead here. Okay, hold on. Let's go back here. Go back. So these 2 slides, I apologize if you want to just combine the information. This information is very basic. It just tells you where you would find the immunoglobulins, and the next slide tells you a little bit more about what they do. So IgGs are going to be one of your major antibodies, and they're found in the plasma and the tissue fluids. And as you're going to see, they're going to be the ones that are going to tag bacteria, viruses, and certain toxins for destruction. And they can also activate something called complement, which we'll talk about in just a minute. IgA are going to be antibodies in your exocrine secretions, meaning, places like your tear ducts and your sweat. IgMs are going to be the antibodies that are inside your plasma. Some of them, again, can attack bacteria, and some of those IgMs are going to be the antibodies that will attack, a blood cell that has the opposite blood type. So, for example, when we talked about if you're type A and you have, the B antibodies in your blood, those B antibodies are IgMs. They're an example of an IgM. Okay. And then IgDs are going to be on the surface of your B cells. You'll see them on the surface of your B cells to recognize, again, foreign invaders. And IgEs are going to be in exocrine secretions as well. And you'll see they work in allergic reactions, and we'll also talk about their role in parasitic infections here in just a minute. Okay, so this is probably the more complete slide on antibodies. And if you want, this slide just tells you where you find them. This slide tells you a little bit more about them. So this is, if you want to combine kind of these two slides as you're making note cards, that would be my suggestion for you. So IgGs are going to be the most abundant antibody in your blood. About 80% of your antibodies are IgGs. And these are going to be the ones that you kind of typically think of when you say the word antibody. These are going to be found in your blood, in your lymph, in your intestines, and they're going to perform most of the antibody functions that we just previously talked about. So again, they're going to perform most of the antibody functions of tagging bacteria, tagging infected cells for destruction. Then we have IgA antibodies, okay? And so these are about 10 to 15% of the antibodies that are found in your body. And you're going to find these in your tears, in your sweat, in your saliva, and in your mucus. And these oftentimes increase, or these are the ones that, decrease during stress. So this is why oftentimes when we say you're stressed out, especially, around final exams, when you're stressed, you're more vulnerable. Your immune system is more vulnerable to picking up infections. This is why, because IgAs are going to decrease significantly during stress. And these are found in, again, areas where you might initially see an invader introduced to the body. So in your tears, and your sweat, and your saliva, and your mucus. So again, now you're not killing as many of the bacteria in these areas where your bacteria might be first touching your body. So this provides localized protection against pathogens. IgMs make up only about 5 to 10% of the antibodies in your body, and these are found in your blood and in your lymph as well. And these are the ones that cause agglutination. Okay, so when we talk about the ABO system, where again, if you're type A, and that means that you have again antibodies for B, you have the B antibodies. If you're type A, right? And if those B antibodies are an example of an IgM. And so again, let's say if you were given type B blood, these B antibodies would bind the B antigens on the B blood cells and cause clumping or agglutination. Okay? So those are your IgMs. IgDs make up a very small percentage, 0.2%, again, of your antibodies. And remember, these are going to be expressed on your B cells, and they're responsible again for the activation of your B cells. So these are going to be the antibodies that are basically on your B cells. They're very specific for that certain type of antigen, and now that's what activates the B cell, is binding antigen to that IgD receptor on the B cell. So that's what your IgDs are. And then your IgEs, and if this happened, just to give you an idea, let's say this happened, here's your IgD antibody, bound again, an antigen, and it activates this B cell, then the B cell undergoes cell division and produces a bunch of plasma cells, and then the plasma cells are going to release IgGs, okay, into the blood. So then your IgE makes up just 0.1%. And it's involved in allergic reactions and also in parasitic worm infections. So it's an antibody that's specific for, again, antigens, like maybe you took in some pollen and you were allergic to it. Okay, your IgE antibodies are going to, again, be responsible for some of your immune response. Or, again, if you have some type of, parasitic worm infection. And believe it or not, we all take in parasitic worms, as much as you might not like to know about it. We all have little round worms inside our system as we take them in foods and things like that. And so our IgEs will actually, again, bind to those worms. So it's interesting, hint, if you were to see a question, remember that eosinophils oftentimes are elevated, again, because eosinophils are one of the white blood cells that actually help to take care of parasites. They engulf parasites. So our eosinophils are oftentimes high, and our IgEs are high if we have a parasitic worm infection. So hint, hint, this could make a very easy question. I could say, a patient presents with elevated eosinophils and elevated IgEs. What are they most likely to be infected with? They're most likely to be infected with a parasitic worm. So that's one of the ways that they can test to see if you're infected with a parasitic worm. Okay, so this is kind of cool. This shows you, again, what the different antibodies look like. So your IgGs, your IgAs, and your tears and sweat and things like that. Again, your IgEs that we just talked about that are released, again, for parasitic infections and allergic reactions. Your IgDs that are on the surface of your B cells. They're going to look very much like your IgGs. And then your IgMs. And this is cool. Check this out, guys. So this is how you can see your IgMs, remember, are the ones that are associated with agglutination. So let's say, for example, that this was a B antibody. And this was in a person that has type A blood. So let's say this person had type A blood, okay? And they have these B antibodies running around in their system. And all of a sudden the person was given a transfusion with the wrong blood type. They were given a transfusion with B blood or B blood cells. And these B blood cells are going to present the appropriate antigen. Okay, oops, I drew that. Let me go back and draw this again. So they'll be presented with these B blood cells, okay, that are now presenting antigen that's going to be bound up by that antibody, because this is a B antibody. And so what's cool about this is that if you picture this IgM antibody, you can see why they cause agglutination. Because check this out. Look at how many antigen binding sites they have. So look at this. This one here can bind, okay, 1, 2, 3, 4, 5 blood cells here, okay? And this is how they cause clumping. This is right here, the IgM antibody, okay? The structure of it is what causes the clumping. So I thought that was really cool. You can see now this relates with back to the blood chapters, that you can see why we get agglutination. It's because of these antibodies and the shape of this IgM antibody and how it can bind multiple blood cells there. Okay, other antibody actions. Antibodies, again, are going to react to antigens in a couple of different ways. The first one is the one that we've really talked about. And one is that it will simply neutralize whatever that invader is. So, for example, if it's a bacterial cell, here's my bacterial cell, okay, and it's got its little antigen on here. Now, if antibodies are released and antibodies bind to this bacteria, now this bacteria can no longer enter other cells and infect other cells. Okay, so it's neutralized. Okay, it's also now amplified other immune responses, meaning that it's made it more likely for phagocytes to come in and eat this cell. So now macrophages can come in and engulf, okay, this foreign invader. Okay, so that's another major action of antibodies. They amplify other immune responses, especially they enhance phagocytosis. And then we're going to talk about in just a minute how they activate complement. And antibodies can also, again, cause agglutination or clumping, just like we talked about. So we just talked about how, again, an antibody can, again, cause clumping, especially when we talk about those IgM antibodies back here, okay? These are the ones that especially can cause clumping. And then IgAs, as you can see, because they can bind too, can also cause some clumping as well, okay? So some of those antibodies can cause clumping or agglutination. And then, again, that third response is to amplify other immune responses. So either, again, to call in these phagocytic cells, or they can actually activate a system that we call complement, okay? So if, again, an antibody binds to an antigen, just the presence of that antibody bound to the antigen will basically activate a bunch of these proteins, complement proteins, to be released. And when these complement proteins are released, They will then enhance phagocytosis, so they'll call in the phagocytes, okay? Some of these complement proteins will call in phagocytes to that area. They'll attract the phagocytes into that area. That's something that we call chemotaxis. So basically these proteins will call in and say, hey, phagocytes, come over here. This is the bacteria that you need to engulf. This is the foreign invader. And then they can even call in certain proteins that will cause the membranes of that foreign cell to lyse. So again, some of these complement proteins that are stimulated by just the binding of antibody to antigen will actually cause the cell membrane of the bacteria to lyse. So that's complement. Okay. They can also do a couple of other things, by like altering the molecular structure of viruses, but these are the biggies here. Okay, so now this is kind of cool because this shows you why we get vaccines. So if you look here, this is just showing you our primary and our secondary immune responses. Here, if you look, this would be the first time that you were exposed to something, okay? And this is just showing you two different types of antibodies, IgMs and IgGs. Now, IgMs are a little bit different because remember that those are going to be a response, again, inside your blood. We have a certain number of IgM antibodies running around in our blood versus IgGs, which are going to be produced by your plasma cells, okay? So let's talk primarily about your IgGs. Okay, so what happens here is if you look in your primary immune response, that's your first exposure, okay, you're going to see that you produce a lower antibody titer than your secondary exposure, right? Big time. And that's because what happens here, remember, is when you're first exposed, then those B cells, okay, have to divide and produce lots of plasma cells, and then they produce lots of memory cells. Okay. Now, what's nice now is that we have more of these memory cells running around our immune system. So what that means is we have more cells if we were exposed the second time. Okay. On our second exposure, we have more plasma cells that can quickly undergo division and produce more antibodies, okay? So here, in this example, here you just have this one B cell that was activated. Now we have all these B memory cells for the second time that we're exposed. And now all these B memory cells, okay, are going to be producing antibodies and spitting out antibodies out of these plasma cells versus, again, just this one B cell producing these plasma cells. So all of these memory cells now can undergo cell division and produce plasma cells and produce tons of antibody. So what this means is all of these extra memory cells that we have, okay, are going to allow us to produce a lot more antibody, and we're actually going to produce them even faster, okay, so then after our first exposure. So what this means is in our secondary immune response, okay, we produce more antibodies and we produce them faster. So this is why you got vaccines, right? This is the whole reason why I get a flu vaccine every year. Is because if I get that flu vaccine, then my chances of really getting sick from the flu are much less likely because I have all these memory cells running around my system, okay? And they're going to produce that secondary response, which is, again, I'm going to produce a higher antibody titer much faster. Okay, so the last couple slides here are just going to talk about the different types of immunity, and we're going to classify immunity based on four major kind of categories. Our first category is, of course, our natural acquired active immunity. And what that means is we acquired this immunity by being infected by that organism, okay? So let's say you got the flu virus, okay? And so then you now obtain some immunity because now you have those plasma cells, those memory B cells and you get memory T cells too. So you have memory B cells and memory T cells are running around your system now. And so you naturally acquire that immunity for those memory B cells and memory T cells by becoming infected with the flu virus the first time. So the second time you're exposed, you're much less likely to get sick. That's naturally acquired active immunity. Then there's artificially acquired active immunity. And this occurs through the use of vaccines. So again, you're injected with antigen. Oftentimes you're injected with either the antigen or just the dead virus or an altered virus that is not going to, again, infect your cells and cause disease. And so what happens then is that activates the immune system, though, and because they have the same antigens as, you know, your live virus would, then you're going to produce the memory T cells and the memory B cells. But the beauty is that you produce those memory B cells and T cells without becoming ill, right? So again, this is the idea behind vaccines. They're artificially acquired, but they're still active immunity because you still, again, are producing those memory T cells and B cells based on the introduction of antigen. And then there are two types of passive immunity. And passive immunity is acquired by taking in not the antigen, but taking in the antibody. So there's artificially acquired passive immunity, and that involves actually the injection of gamma globulins, IgGs, containing antibodies that are short-lived. So basically this is from injecting antibodies. And my example for you of this is going to be my husband's uncle actually runs a business based off of artificially acquired passive immunity. And what he does is he has a bunch of horses, and what he does with these horses is he vaccinates them regularly so that they produce lots of IgGs to certain types of horse viruses and certain types of horse bacterial infections. And so then what he does is basically he does just like when you go to donate plasma, he has a machine that it's actually the same type of machine that he does, and he removes the plasma with the IgGs in them because our IgGs are going to be in the plasma. Remember, they're one of our plasma proteins. And so he'll remove those IgGs in the plasma, and then what he does is he sells the plasma to especially horse breeders of race horses and things like that. And those are individuals that want to make sure that their foals don't get sick with certain diseases. So they'll actually give that plasma with all these IgGs, with all these antibodies for certain diseases to their foals to ensure that their foals don't get sick. So that's an example of artificially acquired passive immunity. And then we have, you of course can have naturally acquired passive immunity, which is the way that we naturally pick up antibodies from mom as mom passes antibodies to the fetus. But remember that these antibodies are short-lived in both cases. So these antibodies, again, are going to be broken down eventually, and they're not stimulating the memory B cell and T cell response here. So this is a much more short-lived form of immunity, okay? So again, vaccine is a long-term way to provide that immunity. But so back to naturally acquired passive immunity. Examples of this would be, again, a mother passes it on to the fetus, antibodies to the fetus through the bloodstream. Also, a breastfeeding mom will pass on antibodies to an infant. And so, again, that would be a natural way. And one of the reasons why the foals are given the plasma is that oftentimes they're weaned early from the mother. And so if they're weaned early from the mother, then they're not getting the naturally acquired passive immunity from mom. So then they go ahead and give them the artificially acquired passive immunity by giving them the plasma. But again, breastfeeding is definitely a way, and this is why now for a while, for some reason, in the United States, breastfeeding was not encouraged, and we've now gone full circle, and we're now encouraging breastfeeding again in the hospitals. And the reason that we're encouraging breastfeeding again is because of this naturally acquired passive immunity. You're passing on antibodies to your very vulnerable infant who has an underdeveloped immune system, and that gives them this naturally acquired passive immunity. All right, the last thing I think that we're going to talk about is allergic reactions, and we're going to say very basic here. Allergic reactions to allergens are excessive immune responses, okay? And so these are not normal immune responses. These are excessive immune responses, and they can sometimes even lead to tissue damage. For example, if you have a super, allergen that you're producing an immune response to. So again, keep in mind that when we talk about allergic reactions are excessive immune responses. They're not part of our normal immune response. During allergic reactions, we have certain cells called mast cells that are going to release histamine and leukotrienes. And these two chemical signals are going to induce inflammation. Okay? Certain allergy mediators then can actually flood the body, and these certain allergy mediators can sometimes result in anaphylactic shock, which is obviously a much more severe form of an immediate reaction to an allergy. So again, that's not always going to happen, and oftentimes, again, depends on that individual's type of immune response to that allergen. Some people may just be slightly allergic to cats, and so they might produce a more minimal immune response, and some people might be really allergic to cats and may produce a much larger, much more excessive immune response. And again, if it's excessive enough, like some people are allergic to bee stings, they could actually put them into anaphylactic shock. Okay, autoimmunity also is one of my last topics. Sorry, I forgot about this one. So autoimmunity is important, and I used to work with multiple sclerosis, which is an autoimmune disease. And in certain autoimmune disorders, the immune system manufactures antibodies against some of its own antigens. So what happens in an autoimmune disorder is the body is attacking some of its own self-antigens. For some reason, it's recognizing them as foreign, and this is why it's called an autoimmune disorder. So for example, in multiple sclerosis patients, I worked with multiple sclerosis. For some reason, their immune system recognized the myelin sheaths around their axons as foreign, and so their own immune system would attack the myelin sheaths, especially the cells like the Schwann cells and the oligodendrocytes that were producing that myelin. And so again, that's an example of an autoimmune disorder. And obviously there are lots of autoimmune disorders out there, and many of them, again, can be very detrimental to a person's health. So, autoimmune diseases are definitely an interesting topic. And unfortunately, fairly prevalent out there. Also, transplantation. Obviously, if you transplant a tissue into someone's body, then their body is going to recognize those antigens as foreign and produce an immune response to them as well. So oftentimes, when an individual is undergoing a certain transplant, they actually have to be given immunosuppressants to suppress their immune response so that they don't reject that tissue. Okay, so I think that is it for today's lecture. So like always, if you have any questions, please feel free to write into me. Please write in your questions. I haven't been receiving that many questions lately, but I check the discussion board regularly. So if you have any questions on this material, then please ask. And this will be the end of the material for exam 3.